analogy for this, which I find a little bit useful. Fading into view now, not that long ago, scientists, biologists, chemists were perplexed about how science could explain life. Life seemed to be something beyond what could be explained in terms of mechanisms. This, of course, led to vitalism, this idea that we needed an enormous power to spark life. Now, we don't think of it this way at all. There's no need for that hypothesis of a special source. And my hope and belief is that the same thing will apply to consciousness. Instead of treating it as one big, scary mystery in need of one Hundinger component of the solution, by examining its properties, separating them out, explaining them in terms, we will resolve the hard problem of consciousness just as the hard problem of life was resolved in the past. Now, life is not the same thing as consciousness. This is certainly very true. I think one of the most important differences between life and consciousness, you can't really put a conscious experience on the table and look at it. Conscious experiences are intrinsically subjective. They exist for the organism. This is the problem. It makes consciousness hard to study. How do you get the right kind of data? It doesn't make a science of consciousness impossible. Sometimes it's just means it's quite challenging. You can still get data about what people's experiences are like, and you can try to predict control. So just as in the study of life, people identified different properties that living systems have, things like reproduction, homeostasis, metabolism. We can do the same thing with consciousness. It's not just one big thing. We can think of it in several different ways. And the ways that in the book and in my work with my lab in Sussex focus on different aspects. There's conscious level. How conscious are you when you're conscious? Broadly, the difference between being under anesthesia and being awake and aware. Then there's conscious context. When you're conscious of something. Right now you're conscious of my voice and of this slightly strange experience of being surrounded by lots of people. Feeling of your body and contents of your experience. And especially the last one is related to a very important, for many of us, and certainly for me, the most essential aspect of conscious content, which is the experience of being yourself. The experience of being you. So, let's start. I'm going to rattle through two things about each of these domains of consciousness. And we'll start with conscious level. What in the brain explains the difference between being awake and aware and just being a chunk of living meat without anything going on? The first thing to say is that conscious level is not the same thing as wakefulness. Now, normally they go together, right? You can go to sleep and you can use consciousness of it, wake up, lie down. But there are occasions where the two dissociate. When you are asleep and you're having dreams, which you're conscious when you're dreaming, dreams conscious, but you're asleep. And on the other side, there are states such as the vegetative state or the unaware wakeful state, where people after severe brain injury go through cycles and wake, but there doesn't seem to be anything going on. There doesn't seem to be anybody at home. There seems to be no consciousness. Consciousness is not the same thing. So what we need to do is ask what in the brain goes along with consciousness and not just with wakefulness. And among many different approaches to this, and to cut a long story very short, I'll just tell you about one approach that we've been following in the lab and now with colleagues in Cambridge and London who are here down in Pedro and Fernando and so on, Tristan. We use this measure called length-of-sieve complexity. This is basically, it's very simple really, it's really the same thing that takes a digital photo and compresses it into a short file that you can send by email. And it does this basically by counting the number of repeated patterns in a photo or in any kind of data. And it looks at how many patterns I'll ask questions about that later, complicating the problem. But basically just saying how many different combinations of ones and zeros are there in any data set. Count the number of those, the library that you need to recreate the original, and that's the length of zip complexity. Or you can think of it as the higher it is, the more diverse, the more patterns there are in the data. And if you apply this measure to data from the brain rather than to a photo, you see some interesting things. This is where anesthesia, the complexity level goes down when you lose consciousness. Your brain becomes more regular, more predictable as you lose consciousness. The same happens in sleep. This is another data set where colleagues recorded from electrodes planted deep into the brain. What's interesting here, you can see here that wakeful rest and rapid eye movement, REM sleep, are basically the same pattern there, which means that this measure is the same for the stage of sleep when you're likely to be conscious and dreaming than when you're awake and aware. But in other stages of sleep, it's much lower. And then finally, and what was quite a surprising finding, when we apply the same measure, surprising material, to data from people in the psychedelic state, whether it was LSD here, and also psilocybin, ketamine, it goes the other way. The brain becomes more diverse, more unpredictable in the psychedelic state. This was often written up in the media as scientists discover evidence for a higher state of consciousness, which is not what I'm trying to say. There's something measurable in the brain dynamic state that is characteristic of the psychedelic state. Now these are a first step towards tracking the neural fingerprints of conscious level in the brain, but it doesn't necessarily explain anything about why that should be the case. And so to bring you kind of up to where we are with this sort of work, trying to develop measures based on this idea that actually give the principal reason for why they should go along with consciousness. And this is based back in work that actually first really inspired me to do consciousness science as a career, try and get into it anyway. Back in the late 1990s, I was doing my PhD and I read these papers by Gerald Edelman, who became my boss in San Diego, and Yuya Konomi, one of his proteges, who's now a real leading figure in the field. And they had this idea that every conscious experience that you have has two properties. It's both integrated, it's all of the pieces. Don't go on separately from others. Right now it's all happening in one uniform routine. But it's also very informative. So every conscious experience that you have rules out a simultaneous occurrence of a vast repertoire of alternative possible experiences that you can have. It's very informative. It's reducing the uncertainty in space and all the experiences you can have. These are two very distinctive things about consciousness that don't readily apply to other things about biology that you might want to explain. So we want measures in the brain that reflect these two properties at the same time, that catch the middle ground between information and integration. And this is something we've been working on in other groups too for a number of years now, and this is still at the level of just developing the actual measures. And again, these are the last equations, the only equations we're going to do, and they're not particularly relevant, just to show that there are ways now to write down things that capture this middle ground, and the idea is we can go and test these measures on data from the brain and see how they behave. Right now they don't behave as nicely as the simpler measures. So there's still a lot of work to do, and one of the things that we're moving on to, and this is the last thing I'll say about level, is to build also measures that capture inertia. This is another super interesting, quite controversial, quite difficult to define phenomenon in which a bunch of parts, in this case a flock of starlings, the flock seems in some way to have a life of its own. It seems to be more than the sum of individual birds that make it up. We all recognise when it happens in a situation like this, but it's surprisingly difficult to figure out how you mathematically figure out where and when it's happening in other systems. Like if your neurons are firing in complicated ways, how do you know whether it's sort of like a flock of birds or not? Quite a difficult question to ask. So with Lionel Barnett and Amanda Rosas, we have been trying to develop ways of quantifying that and see if that connects to also the measure. Okay, so this is a whistle stop to work through these things, so I'm going to move quickly on to conscious content. The question of how our demonstration to get down from the heady heights of equations and so on. I don't know if you've seen this before. Yeah, there we go. What I'd like to do is focus your eyes on the black cross at the centre of this image and just try not to move your eyes and try not to blink. Now, without moving it, just how many people see that the magenta patches have disappeared and the green patch is that you can do on Zoom all last year, just like making last week. So now blink and move your eyes and have a look around. Of course, the magenta patches come back. There is no green patch. This is called a light. And what's actually happening here is a few different things. There's something called Troxler fading. When things with indistinct edges are at the periphery of your vision, they tend to just disappear from your experience. There's also apparent motion when things change on and off near each other. The brain infers motion between them. And there's colour opponency. The brain has a colour space, and the opposite in colour space to magenta is green. And the reason for that is kind of interesting because you make magenta by mixing red and blue light. But if you think about the electromagnetic spectrum, red and blue and green is in the middle. Blue on the edges. So if the brain gets red and blue, it's sort of expecting the green cells to be activated too, but they aren't because you're only mixing red and blue. So magenta is sort of not green. So when you're seeing the green after image, what you're really seeing is not green. Okay? Good. So the way to understand that nonsense is that your brain is a prediction machine. There is this gap, you can already see there's a gap between how things are and what you experience. And the idea that I've been running with is that this happens because the brain is always making sense, trying to make sense of essentially noisy and ambiguous and uncertain data. Now imagine being a brain for a second. Imagine that you're stuck inside the bony vault of your skull trying to figure out what's out there in the world. Now there's no sound in the skull, there's no light in the skull, it's dark, it's silent. All you've got to go on are these electrical signals, which are only indirectly related to things in the world, whatever they may be. So perception has to be this process of inference, of best guessing, where the brain combines this soundless, colourless, uncertain sensory information with its prior expectations, its prior knowledge about the way the world really is. And that's what we consciously see. We see the brain's best guess of what's out there. That's a very abstract, high-level description of what's going on. There is another equation down. I forgot that one. What this translates to, you think, what does this actually mean for the brain? It means the brain is doing something called Bayesian inference. Bayesian inference is a very old idea in probability and statistics. It's sort of reasoning under conditions of uncertainty. Everything is uncertain. We can use Bayesian statistics to figure out what might be happening in a pandemic to try and locate the missing submarine. It's reasoning backwards from the effect to what the system is. In this case, the effect are the sensory signals that arise in our ears and the causes of what's out there in the world gave rise to those sensory signals. Now, actually, usually very complicated to do anything right now to a computer, to any machine or anything to do Bayesian inference is really complicated. But one way to do it, one way to at least get an approximation to it, is to do this thing called predictive processing. And this is the idea that this is what the brain is doing when it's making a best guess. And the reason it's doing it is because it's a way of doing Bayesian inference, trying to figure out what causes the environment. And the idea of predictive processing is very simple. It's that you have signals going in both directions in the brain. One direction is from the outside in, from the world deeper into the brain. And the other direction is the other direction, from the inside out, from the brain back out to the centre and back out to the world. And if you take the inside out signals and think of those as the predictions, those are the predictions about what's going on. And the outside in signals, what's coming in, and percolating upwards through the brain. Those are just the prediction errors. Those are just telling the brain the difference between what it's expecting and what it's getting. And by continually trying to minimize these prediction errors, get rid of them, change the figure, get rid of prediction errors. These predictions collectively settle on the brain's best guess of what's out there, and that turns out to be very close to what Bayesian inference is. But beyond that nice mathematical equivalence, there's a real conceptual message here, which I still find quite troubling to think about. I mean, we're so used to thinking that perception is this process of just reading out sensory signals. It seems as though the world pours itself into our minds through the transparent window. But it's the other way around. We perceive it coming from the inside out, and the sensory data is keeping our perceptual best guesses in check. So perceptual content is conveyed by the top-down predictions, and bottom-up sensory signals just convey the prediction errors. So the green errors. Now, if you start asking what's the evidence for any of this kind of proposal, there's some, and we've done some of it in the lab. This is an old study now with a former postdoc of mine at Intel. What we do here is we show different images. So in one eye might see an image of space that gradually appears, and the other eye might initially see this changing pattern of colour that gradually fades away. And if you show them together eyes, the brain, instead of combining them into some sort of net, it starts off with the most dominant, so you just see the changing colours. And then after a while, the space breaks through, and that's what you see. So the prediction you can make is that, well, if perception is conveyed mainly by predictions, by what the brain is expecting, then if you cue the person with the word face to expect a face, then they should see a face more quickly and more accurately than when they're not expecting it. And that's what we see on the data. So people see more accurately and more quickly what they're expecting to see compared to what they're not expecting to see. But that's sort of psychophysics you can do in the lab, and it's fine, but it's not really getting us that close to what our lived experience, rich panorama of perceptual experience is like. So to do that, one of the things we're doing in the group now, I like to call it computational neurophenomenology, and I wish it called it something else. But this is just the idea of using computational models to try and pull this bridge between what's happening in the brain and what our experiences are like. And we're doing this at the moment by focusing on unusual experiences. A good way to understand a system in general is to look at it when you push it and see it's behaving a bit differently from normal. And in particular, in vision, we have this phenomenon of something like hallucination. Now, this is a picture of a face in the cloud. We're familiar with it in some way. We see space in the clouds, we can see space in the the brain is strongly wired to be sensitive to space. And so what's going on here, this suggests the brain has a strong prediction about spaces. So can we then understand, can we try and simulate that somehow, this balance? What happens when you turn up the predictions a bit? And so this is quite old work by Kesuke Suzuki and others in the lab, where we took one of these very well-known neural networks. This is just a neural network that takes images and tells you what's in them. All the information flows from the image upwards. But you can turn it the other way around, and you can fix the output and basically say something like cool, and then run it backwards and change the image. So in that case, what you're doing is you're projecting a prediction into a pre-existing image. And if you do that, we did this in a panoramic video and replayed it through a headset, so people can put a headset on and look around. And what they see is a world confused by dogs and dog parks. And we call this the hallucination. And if you actually look at it through a headset, it's surprisingly compelling. And it's not, some people say it's a little bit tricky here and there, probably not, but it's definitely unusual, and there's something going on. It's not just dogs photoshopped onto this image, there's dogness emerging from the image in a sort of interesting way. And so this is what happens if this is a simulation of what happens if your brain has overly strong predictions. So what we're doing now, just again, with through Sabrina to do, we're sort of taking it further and trying to simulate different kinds of hallucinations. Hallucinations are complicated. With Parkinson's, certain kinds of dementia can have very complex hallucinations, where there's complete human objects. Other kinds of hallucinations are very simple patterns and shapes, and nothing more than that. So we have now a more sophisticated set of neural networks that we wire up in all sorts of different ways. And what we can do is now simulate different kinds of hallucinations. So what you see here, each of these images is like a starting point. A bird, a mushroom, a lamp, a volcano, and a thing. And the second row down, it's a bit blurry and that's deliberate. What we're simulating there is hallucinations that happen when people lose central vision. It happens in something called focal syndrome. And if we run the system, we'll see that we end up with very different kinds of hallucinations in each of these groups. This is now where we can just get a bit more specific about the mechanisms that underlie different perceptual experiences. That is getting us a bit closer to understanding how experience is constructed in our own life. We're also closing the loop on this by finding people with these conditions who experience these hallucinations in real life, and match the output of the model to what they encounter in their daily lives, so we can calibrate So this way of thinking leads to the idea that hallucination is a kind of uncontrolled perception. It's what happens when the normal business of inferring what's out there, balancing perceptual predictions against prediction errors, goes off the rails. And in turn, what that means for me anyway, and I first heard this phrase from Chris Frith, you heard it from somebody else, and let me know find out where it really came from, is that perception is a kind of controlled hallucination. Now, this is often misinterpreted as saying that perception is a claim that perception has nothing to do with the outside world, that everything is made up. Go and stand in front of a bus and tell me, you still think perception is a hallucination? The control is just as important. The way we perceive things is reined in by the world, and not sorts of interesting and useful ways of doing it. But I like this phrase because it just underlines the continuity. Hallucination is not a completely different thing. It's the same underlying process. It's just the balance between prediction and prediction errors has gone. The brain's best guesses on the world, and people see things that other people don't. Now in the book, I try and take this idea much further than just seeing dogs on such as campus or seeing a face more quickly. And the larger claim here is that this way of thinking applies to all of our experiences, all of the time. It applies to colour, sound, touch, taste, but also more fundamental things like a sense of time and duration, whether a visual impression feels like an object or not. And ultimately, whether things somehow seem real. Even that is an aspect of perceptual experience that we have of what we perceive really exists. That too, just like change, is another aspect of perception. Gives a feeling of like a real world experience on one of the few summer days in writing. And take these ideas and turn them into the self. And to recognize that the self is also a form of perception. Now it's quite natural to think maybe that the self is the thing that does the perceiving, that you have a world out there, sensory data comes from the world, there's a self that's somehow the recipient of this data that forms perceptions that reflect the way the world is. But it's not really like that. I don't think it's like that at all. The self itself is another form of perception. It's another kind of controlled hallucination, but a very special one. It's the experience of being you. first implication of this, the new thing is just the self is not one thing. It's another division between how things seem and how things are. So it might seem suffering with mental illness of some sort, something that the experience of being a self is universal. Just you, and you go along from place to place, or Zoom meeting to Zoom meeting, and it's the same you that's there. But there are many ways in which we all experience being a self. There's the bodily self, the experience of being a body, just being alive and identified with this thing in the world, this object that is your body. There's a place where your body ends and the rest of the world begins. There's the experience of perceiving the world from a physical perspective. I know this can go away because people report things like out-of-body experiences where they see themselves from an external perspective. There's the volition of self, the experience of making actions, deciding to do something, being the cause of things that happen in the world. People often think this free will is related to and then finally there's higher levels of self. There's something like the narrative self, where self becomes associated with a set of memories, with a persistent identity over time. And then there's the social self. This thing I think many of us have been missing, this part of us that depends on being with others. Part of what it is to me is refracted through the mind of people I know interact. And all of these different aspects of self all seem to be bound together, all seem to be part of a piece, but they can come apart in all sorts of ways. And we know this from, especially I think the writing that people like Oliver Sacks have beautiful illustrations of what happens when one part of the self is changed, but the other part might remain. In the book I talk about a person called Clive Wearing. Clive Wearing is a musicologist, and he had a very, very severe form of amnesia, a very severe brain disease, which led to one of the most dramatic and profound amnesias, losses of memory that's ever been recorded. He could not remember anything new. He lived in what was called a permanent present tense of about 10 seconds for years, in his 80s, in the 80s. But the rest of him is still there. And I never met him, I knew this secondhand, but there was this famous event where he went back to the choir that he used to direct, and he was able to direct the music with a fluency that made him seem whole again. I'm going to talk a little bit about the body itself. This is where I've done, I think, the most, my interest is focused on groups. This is the experience of both identifying with this object in the world that is your body, and a more basic experience of just being a living organism without shape or form. And the idea, of course, is that these experiences having and being a body are conceptual constructions, are brain-based best guesses, just like everything else. And there's a very famous illustration of this, which I think many of you might have seen, called the rubber hand illusion. What happens in the rubber hand illusion is that a volunteer, a guy in blue, hides his real hand is hidden behind a partition. You can't see it. And a fake rubber hand is placed in front of him, which you can see. Then the experimenter, the guy in green, takes two paintbrushes and strokes both hands simultaneously. And what happens for many people, not for everybody, but what happens for many people when you do this, is that they report this somewhat uncanny feeling that the fake hand is sort of something maybe a bit like a part of their body. Not completely, but maybe a little bit. And if you stroke them out of time, that doesn't happen. So this is in fact, I use this in a lot of talks that I used to, as a fantastic example of the brain making the best guess about what is and what is not its body. You can just see it. There, stroking it, and there's a way it's set so it actually works, which is to use a custom implement. So there's a sort of nice story here where you can think that, yes, the brain is getting evidence that all the sensory information is coming, I here the brain is seeing a hand that's feeling touch, the hand looks roughly like a hand where it should be, so it's making its bad guess. Now this might be part of the story, but one of the fascinating lines of work that's been happening at Sussex, especially led by my colleagues Pete Lush and Zoltan Dienes, is that that's not the whole story. In fact, that might not be part of the story at all. Embarked on the world's longest rubber hand study, probably a couple of years ago, doing the rubber hand illusion on about 400 undergraduates in these kind of rubber hand factories they set up at Sussex at the beginning of the pre-pandemic academic year. So we used the rubber hand illusion on a whole bunch of people, but at the same time also measured something called suggestibility. Now you've all heard of hypnosis. It isn't just stage. We all differ in how suggestible we are. There's a tendency we all have that if somebody encourages us to do something or to expect something to happen, or to experience something, if we're highly suggestible, we'll be more to do that thing or have that experience. And it's a very stable trait. And it turns out that this correlates quite strongly with the rubber hand illusion. So you've got that line to the graph in the paper as it goes up that way. So basically, the more suggestible you are, the stronger the rubber hand illusion is. Now there's two colour bars here because that's comparing when you stroke in time and out of time. You might say, aha, but there's still a big difference. Well, yes, but in fact, and this is other important things, people expect there to be a difference. If you just tell people what you're going to do in the rubber hand illusion, I'm going to stroke your hand right in time and out of time, what do you expect to feel? People know what they should experience. And so it's actually no surprise there's always a difference between them. In psychology, this is a very old issue of demand characteristics that we always worry about. How do we control what people expect to happen? Because people doing psychology experiments are really annoying. They always try to figure out what you want to happen and either do that or more likely you won't. But all this is part of the same thing, saying that what we experience is still this construction, but there are many different factors now. It's not just the sensory data, there's all the things that the brain is bringing to that context to experience. Okay, now in the last bit of the talk, I want to talk about not the experience of the body as an object, but the experience of just being a body. Basic basal ground state of what it feels like to be alive. I call it the idea of being a beast machine, which I'll explain why in a little bit. So having a body is one thing. And by the way, there is something really distinctive about that. In other cases, we know from lots of neurological cases, there are cases like there's a thing called somatoparaphrenia, where people who have a specific kind of brain damage, they will experience their real limb, they still have maybe a right arm, they will experience it as belonging to somebody else. And that experience is held really powerfully. They will try and throw themselves out of bed, they just do not believe it's their arm, even when you point out that it's drawn to their shoulder. So that's the sort of strength of experience that you never get from so this body ownership thing is fundamental stuff. But there's also this experience of just being a living organism. And we don't think about this quite so much. We slightly used to think about this so much. It's really often what being is all about. And it highlights another form of perception. We're very used to thinking of perception as something that's mainly about the outside world. You have your eyes, you can listen, figuring out what's out there in the world. But there's a whole variety of perception called interoception, and this is about the brain perceiving and regulating the interior of the body. Think about again what it's like to be a brain. It's got no direct access to anything, whether it's in the world or in the body. Yes, it's sensory. Don't come and label like, oh, I'm the heart, I'm the kidneys. It doesn't work like that. So this concept of interoception refers to all the various ways that the brain senses its own physiological conditions. And if you think about it, that's fundamentally what brains need to do. They're not there to write poetry, have nice habits, or figure out marketing faced with the same problem. There's no direct access, so this perception of the body from the inside follows the same principles. I think is going on that it follows these principles of prediction and prediction error. But now the dance between prediction and prediction error is unfolding primarily within the body itself. This is this concept of interoception. And there's one crucial difference between perceiving the body from within and perceiving things out there in the world, which is that interoceptive prediction is perception about the body. They're not about finding things out, they're much more about controlling things. So what matters about the state of my body from the brain perspective is not where my lungs and kidneys are, and what shape they are, what colour they might be, but how well they're doing their job at keeping me alive now and in the future. Whereas when I perceive the outside world, ultimately that's what it's for too. My vision work is ultimately the service of me staying alive. But more immediately, I need to know where things are. Is it coming towards me? Is it going away? Is it going So interoceptive predictions are basically doing a fundamentally different thing. And I think that underlies why the perceptions of the body from within feel very different, have a very different character than experience of the world around us. When we think about experience of the world around us, it's objects and places and people and things and stuff. But causing body experiences, emotions, moods, things feel good or bad. And we're likely to be good in the future, and we're likely to be bad in the future. That's a pretty limited view of emotion, but it kind of comes down to that. Things are ultimately good or bad in some way. Now this idea brings together, and in the book brings together a lot of different ways of thinking about the brain. This is a very, I think, hugely exciting but much overlooked tradition of cybernetics. Back in the 1950s, when people were first thinking about computers, they weren't only thinking about computers to play chess or to play Go. There was another completely integrated at the time line of thinking about systems that could control things like just after the war, so things like radar and guiding missiles were critical. This idea that a good control system has to have tightly coupled feedback, it has to be very sensitive to error prediction. It also has to have a model of the system that's controlling. It has to be able to make predictions about it. It needs to know what's going to happen to it in the future. So the idea is that any control system worth its salt in a complex situation is going to involve this process of prediction. Now, your central heating system at home, or air conditioning system in America, can work very simply. Like if it's too cold, it comes on. If it's too hot, it turns on. But when it comes to something like the body, it's not enough to do that. You don't want to wait until your heart rate is gone through the roof before you decide to try and dilate your blood vessels or do some other thing. Your brain decides to do that. You have to predict in advance in order to keep things stable. In physiology, this is called allostasis, achieving stability through change. So this idea has a very long provenance that motivates this idea of predictive perception from the perspective that that's how the brain controls things. This reaches, I think, it's sort of the most fully elaborate and beautiful analogy in this idea of the three energy principle, which is developed by Carl Friston. So here, and this is the idea that organisms, especially organisms, are basically things. Anything that counts as a thing maintains itself over time, doesn't dissipate into nothing. An organism, or things that maintain themselves over time, just think about like a fish. A fish swimming around in the water remains being a fish because it's in the water. If you take a fish out of water and put it on a table, it's mush after a while. Living systems in particular maintain themselves in states they expect to be in the long run, to try and stay in a very small repertoire of possible states they could inhabit. I am not turning into mush, and my brain's job is basically to try and keep myself in this very small state of all the possible states I could be in, most of which would be mush. And it turns out this very basic imperative being a thing gives rise to this rich machinery of prediction and prediction error. So just to summarize this last bit, I think I've already hinted at this, the reason this true relevance of consciousness is that these different kinds of prediction explain why different perceptions are the way they are. Visual perception underpins visual prediction, underpins visual perception. You see objects in places because that's what matters. That's what visual predictions are all about. Interoceptive predictions underpin embodied experience, how well things are going. Those have a very different character. And this brings us back to where we started, this idea of self-change mind. I experience myself as relatively stable over time, even given good photographs. Why is this? Why is it happening? I think one reason for this is that if predictive perceptions in my brain are geared towards keeping me alive, for them to work well, it basically expects me to stay stable. It expects me to not change over time. The stability of the cell becomes a self-fulfilling prediction. Because to remain stable, to remain alive, to predict myself to not change. It's subjective to mind to change myself for this reason. And this applies not only this is conjecture, I'm not trying to say this like, oh, we've got this, trying to prove this. I think it makes sense and this applies not only to these basic experiences of being a body, but to all our experiences of being a self. There's something very useful if the brain is trying to keep the body going for a long time. So experience itself as unified, as stable, is a good way to make sure that what the organism does is always drawn to that as the most likely outcome. So the subjective stability of the self as a whole becomes a self-fulfilling prediction. We predict ourselves into existence. So let me just wrap up where we are. What we consciously see depends on the brain's best guess about the causes of sensory signals. The hand illusion and other experiments of this sort applies not just to the world around us, and that the predictions that matter for the body are not just predictions about the body and an object, but about controlling and regulating the internal physiological state of the body. This means to me that all our perceptions, they're all grounded fundamentally in this imperative to stay alive. These predictive mechanisms that arose over evolution and development all have their origin in this primary imperative to keep the body going, to keep the world around us and ourselves moving through and because of our living bodies. This means, again, that consciousness actually is really quite closely related to life. It's not just an historical analogy anymore. There's a close relationship between being alive and being aware. And this is where we can finally bring up Bacon's consciousness. But it's not because this is dualism here. It's more because of what he said about the relationship between life and mind. He said about other animals, and I always feel bad about giving him credit because he's living at difficult times and he's brilliant, but he did say this about animals. Without mind to direct their bodily movements, animals are unthinking, unfeeling machines that move like that. So being alive is irrelevant to the rational conscious mind that humans have. Now I think it's entirely the other way around. Conscious selfhood arises because of, not in spite of, our human nature. Now I'll finish in the last couple of minutes with just a couple of implications from this that are drawn out in more detail. One is free will. Free will is something that we can talk about mysteriously, but free will is another level of mystery altogether. It's the perception of being, the perception of making actions that appears to come from within. When we experience the free will of action, it feels as though we're making something happen that needn't have happened, or I could have done differently. Or the world, I'm intervening in the universe in some way. That doesn't mean that that's actually true. In the same way that when I experience red, it doesn't mean that the redness is really there. It means that my brain's way of making that sense of what's going on. Voluntary actions are real. That have many internal causes, but there's no need to postulate some uncaused cause that swoops in from wherever and changes the causal structure of the universe. That is nonsense. The second implication is that we all see the world differently. Now we know this a little bit in treatments. You might say somebody with psychosis, somebody with synesthesia or something that experiences colour, all experience the world differently. And that difference is often unseen to us, hidden. The same words that describe the same thing. But those things as they appear to us are going to be subtly different. We inhabit our own unique inner universe. And beyond the human, there's a vast space of other minds. I spent a week with octopuses back in 2009, which is one of the most amazing weeks of my scientific life, and I write about it. When you spend time with octopuses, you realize the particular way that we experience being itself doesn't apply to a creature as alien as the octopus can taste with its suckers and its tongue almost like sometimes animals who can inject the portion in blood. Very strange. And finally, there's the prospect of intelligent machines. Some people think that as AI continues to get smarter and smarter, there'll just come a point where the lights come on and it becomes conscious. Now I don't think this is true at all. If consciousness is tightly bound up with being alive, that's what matters. Consciousness is not the same thing as intelligence. That's part of our human sense of universe where we think we're intelligent and consciousness must go together. You don't have to be sentient to suffer. No, you don't have to be smart to suffer. But you really have to be alive. I love this Atomic Magnet explores these ideas about when we infer consciousness. So if we are worried about creating consciousness artificially, perhaps we should be more worried about things like brain organoids, this new neurotechnology where people are growing brain-like structures in a dish. This is rapidly advancing, and there was a paper that I just saw the other day with a conscious organoid here. I didn't actually do that. Messing around with keyboard, but this organoid actually has light-sensitive pits, the beginnings of eyes, and the neurons within it. Now, these are still relatively simple, but they're made out of the same stuff. They're not made out of silicon. So one whole big unknown about what it takes to be conscious just isn't there anymore. With my colleague Tim Bain and my telemedia, we've written about the prospect of islands of awareness. There's another case with Michael Carter who's here as well, where we think about patients who basically have had operations that leave a hemisphere of their brain alive, disconnected within the body. And on that cheery note, I think it sends a core message, which is that we perceive the world around us and our current within it with truth and because of our living God. And before closing, I would really like just to thank this, of course, the way the work that's happened in the lab has been done by other people, and I've been so fortunate to work with a brilliant lab over the last 10 years at Suffolk. But it's also shaped the way I think about everything too. It's not just the experiments, the ideas that all come as a collective. So I want to thank everybody I've worked with. And the last thing I'll say is that I know it's a bit unusual, but the book is dedicated to my parents. So I just wanted to dedicate this talk to a former PhD student of mine, Paul Shorley. Paul died in 2013, shortly after finishing his thesis, and though some of you here might remember him a bit, he was a lovely man and a brilliant intellect too, and I just sadly miss his memory alive. And I'll leave you with a summary of everything, and thank you very much. give rise, alter brain dynamics as a whole that explains psychedelic experience. The other thing that I think is fascinating about psychedelics is that people who take psychedelics and then ask questions about whether they think consciousness is a material phenomenon that's identical to or generated by the brain somehow, or whether they think it's some immaterial stuff. I would have thought that taking psychedelics would lead you to be more materialist. Like you do something to the material of the brain and your experience changes. Like, really good evidence to me that consciousness comes from the brain. But there are other people who draw the entirely opposite conclusion. Because they have all these weird experiences where you go, oh wow, there's something much more than my brain involved. And so there's this whole second level of conclusion. Because I think it's starting to think where do the models using these predictions come from? It's a good question. They can come from, you can think of them over varying timescales, like some of the models you might have encoded in our brains are very, very hardwired, and they may inherit from evolution. And just as we inherited the more obvious features of our biology, if things in the environment are sufficiently stable, then the brain can learn encoded models over evolutionary time. To the extent you might not even recognize them as predictions anymore, they're just hardwired and stable. Like you can think of, for instance, that the retina is organized spatially as a sort of prediction about the spatial structure of the visual environment, and then it becomes a bit nonsensical about that as a prediction. But when things change, then you think, okay, there's a purpose to having an actual predictive model that can change the predictions. Where do those come from? Well, the machinery will come from evolution, but then there's development as well. Every organism develops and exposed sensory data during its development. Some of them are learned. We can learn to see things differently over periods of days, weeks, hours. And some of them are updated then on the fly. But so there's no single place, and there's no single mechanism that you say is the source of all prediction. I don't think they necessarily do. And I also think that the predictions are not to be assessed necessarily by their accuracy. The purpose of perception, whether it's of the world or itself, is not to figure out with maximum accuracy what's actually out there. That wouldn't be that stuff that's out there isn't relevant to the brain. Colour is not an accurate representation of the outside world. There is no objective colour outside. The colour is the place where the brain and the universe is. Colour is invented by the brain to help guide our behaviour. So it's not, if we were perceiving colour, we wouldn't perceive colour. When you talk about self-change blindness, change the text, right? They think to react to changing environment. The brain is extremely good at it. And yet, there seems to be a superficial conundrum there, which is that we have this consistent within our own existence which refuses change. But doesn't that run contrary to the notion that we need to be very good at spotting change in our local environment? I think it just shows that whether we perceive change or not depends on the utility of perceiving that change and on the prior expectations we have about change. So the changes that we see are changes where things happen very quickly, or they're not. They're to some extent expected. Because notoriously bad. So the things where change lines work in vision is when things change very, very slowly and we're not expecting change to happen. And again, hopefully we know that humans are bad at coping and recognizing changes that happen slowly. talked about red quite a bit. That's my favourite colour in the colour. And that brought up Francis Crick, who was actually, I met him a couple of times in San Diego. He's one of these people who won the Nobel Prize, and then that gave him a license to study consciousness, because consciousness was quite an illegitimate thing to study, because people like Stuart Sutherland were saying nothing worth reading had been written about it. And at the beginning of the rebirth of consciousness science in the 90s, there was, I think, I mean, this is a bit of construction for me in conversations with people like that it's quite pragmatic to study something like vision, because you can control vision very well. We can put people in the lab and control what they consciously see, and we quite control the stimulus, and there's a lot of knowledge about how the visual cortex works. So pragmatic is, okay, let's just look at what happens in the brain when we change the relationship between sensory data and conscious experience. In vision, then we can find the neural correlates of vision. What in the brain is going along with consciousness in particular. And I think that approach was very wise at the time, because just by saying, look, forget about all this complicated philosophy, metaphysics, free will, self-work, woody stuff. If people see something or they don't, something happens in the brain or it doesn't. We can agree about that and not worry about the rest. It's a way of getting the foot in the door and bringing, I'm going to mix the metaphors here, bringing consciousness back to the table in mind and brain sciences generally. And it's still going on. So there's a lot of brilliant work trying to refine this, what are the neural correlates. And there's not just one, there are many. There are neural correlates for conscious arousal, for visual experience, other experiences. It's not going to come down to one simple thing. And one way to think of what I'm doing is going beyond correlation tools that just find correlates and stop them. Doesn't explain anything. Try to choose which one is correlated with the water or something like that. One doesn't explain the other. You need to have a mechanism why that correlation should obtain. Question that came in from people watching online, which I found quite interesting. How long does it take a newborn to get enough decent prediction making machine? What we learn from developmental psychology about how consciousness emerges from birth. Yeah, this is so. The problem with developmental psychology in this case, it's like the problem of other animals too. You can't ask a baby, like, please tell me, are you seeing it? It doesn't. As soon as you see the face of it, it's not going to work. But there's clearly a point at which something happens. One of the guesses, and it is a guess from the perspective I've come to take, is that the earliest conscious experiences in development are not going to be about the outside world, they're going to be about the body, they're going to be emotional, they're going to be self-relationships. They're not hugely elaborate self-relationships, they're famous tests called the mirror self-recognition test. So this is really a test of when does full-fledged self-identity emerge, and basically when can a baby or another animal recognize that a mirror image is itself rather than another creature. And human infants don't generally pass the mirror test until about 18 months, though it's now getting closer to use the experimental way, maybe about 12 months. Does this mean that babies are not conscious until 12 months? No, of course it doesn't. It means they just don't have an elaborate developed sense of personal identity. There are some more brain imaging experiments done by my colleagues in Paris, looking for some of the brain signatures that we know in adult humans goes along with conscious perception. And they seem to be there quite early in human infancy. But the problem is the brain is very different in a very young baby. It doesn't have, like in an adult brain, all the connections have this covering of myelin, which helps preserve electrical impulses in the brain circuits. So the infant brain is not myelin, so you can't take what we see in the adult and ask if we see it or not in the infant. It's a very, very hard question. Yeah, I didn't answer it. No, you did brilliantly so as well. I mean, it makes me think about Nagel and his band, and actually that, we don't know what it is, different species, one perhaps animals, two adults, they're not kids, they're not, but that inner experience is inevitable. It's not just inevitable in us sharing our experiences of bed or whatever it is with each other as adults who have full lexicons and describe things. But the mirror test, the geeks have always thought it was a weirdly eccentric test because you do it on gorillas. And gorillas don't like icons.
No, they don't. Yeah.
Show them a mirror, and they're going to beat the shit out of the guy in the mirror. Yeah. Or you do it to a dog where their sensory, their umwelt is defined by smell. Is there a smell mirror? Yeah, there's an olfactory test. Okay. Olfactory test designed for dogs, and cognitive science in dogs is now called Dognition. So cognitive science. That's good. That's very good. Okay, time for more questions. Wait there. Second round. So the question was, perception is useful, it needs to reach its best guesses quite quickly, so it can't take too long to keep exchanging predictions and prediction errors up and down. I think that's exactly right, but I actually think timescales are not fairly compatible with that. So one idea, and this is something there's some evidence for it, but again, it's still pretty open, is that the brain has all these intrinsic rhythms. So there's the alpha rhythm, which is 10 hertz, mainly associated with maybe other things too. So it could be, and that rhythm is orchestrating this exchange of predictions every 100 milliseconds. That's actually fast enough to match. There's nothing really in the phenomenology of perception that means that that's inappropriate. And also, just remember that the fact that the brain might be updating prediction at a particular time scale doesn't mean that our perception either changes or is changing at that time. That's all going on under the hood. Now, it could be that if you're faced with a completely unpredictable situation, then it takes longer for the brain to reach it, to converge. But in fact, there are cases where that's pretty much happening as well. If you a stranger in the fog thing again, sometimes it takes a while before our perception is set. And by the way, audition is much quicker, in a really rapid sense, and that goes along with the idea that actually the circuits in auditory cortex that might be mediating, they operate at a higher frequency and they're less far apart. So, yeah, I don't think it's a massive problem, but it's very interesting.
That's it. Oh.
You have the cortex, which is this big wrinkled surface, where a lot of the neurons and a lot of the action is, and you have all these subcortical areas and various other nuclear areas. And then below everything you've got the brain stem, which is really important for the basic regulatory processes as well. Now, some people write off the brain stem as saying it's just like the on-off switch for a computer. Yeah, you need it to function, but that doesn't mean that the TV program that you're watching isn't in the plug. It's somewhere else. So the question is the brain stem just an on-off switch, or is it where the action is happening, or is it somewhere in between? Mark Soames is on the other extreme, it's where the action is happening. I'm more somewhere in between. There's very, in my view, limited evidence for the brainstem being the sufficient basis for consciousness. And Mark and I would probably disagree about how to interpret that. There are some people who are born basically without at all, and they do exhibit some sorts of behaviours, and there's a lot of debate about how to interpret those behaviours. But I think the brainstem is important. It's likely to shape our conscious experiences. But just there's a wealth of evidence that just ties consciousness to cortical activity patterns when you lose consciousness in anesthesia. The most explanatory fingerprint of that is in what's happening in the cortex. Okay, time's up. I've got one question to end on. And things which are fundamentally unacceptable, and even though they have this a little bit reference the idea qualia, that there's an ineffable quality. I realise this is not a quick question in the slightest, but it's terrible sharing. You need to finish with it. Yeah. There is a point with this, I will get to it. I'm sharing myself. You presumably take the sort of Danic view that all of these questions are empirical in their nature and will be answered in time. What is the experiment that needs to be done? If I give you an infinite grant, give me the grant now, I'll tell you, do not have that power. Alright, I give you 10 billion. I don't know that either. But what do you want to do which chips away at this ineffable problem? Okay, so there is a very well-known philosopher, Dan Dennett, who's been one of my inspirations over the years. I'm never entirely sure whether I'm completely on board with what he's saying. It depends. I still think that there might be a math problem. That there is something. There are two interesting things about consciousness when it comes down. One is it's really hard to get data. The other is maybe it is a completely different kind of thing. Now, my bet is that it isn't, and that it will resolve. But I don't think, in all intellectual honesty, I can claim that right now and say that it certainly is not. What I do think is that by following this real-time approach, even if there is some residue of mystery left, it changes the nature of the problem in richer ways. Like this whole emphasis on the body and the self and regulating it, you think about free will in a completely different way. Whether I solve how the problem seems to work or not. And I think that's really important. You don't necessarily solve the problem yourself. You still get somewhere. You get often somewhere better and different. So if you give me $10 billion, that's pretty good. I'll hire a lot of postdocs. Well, we'll take your application under consideration. Ladies and gentlemen, we are out of time. Alan's going to be outside signing books. Please get it. There are other books which are worth reading on this subject. Really some. But this is, as I said at the beginning, I think this is definitely become a classic and will really set your brain thinking. So please give us Professor Alan.
I'm going to talk today about this basic question, are viruses alive? It's a simple question in the sense that it's only 3 words, but it's actually a very complicated one when you really dig into its meaning, and it's very difficult to answer, and we're still trying to answer it completely, even though scientists have been trying for a very long time to resolve it. Part of the problem with this question is that viruses are pretty new to science. Certainly, viruses are not new to us. People knew, for example, that dogs could become rabid and attack people and they have to be killed. This disease, rabies, was certainly familiar to everyone, but nobody knew there was actually a virus that was growing inside the nerves of these dogs that caused them to have this new behavior. People experienced viruses themselves very personally, in the sense that they might develop a fever, become very sick, start to cough, and might die. And one of these diseases came to be known as influenza. We think of influenza as the name of a virus, but this name was not given to the virus because this name came about, I believe, in the 1500s. This name is Italian for influence, meaning that people thought that they got influenza through the influence of the stars. The concept of a virus was totally lost to them. There was just no way to know. So understanding viruses and what they really are was simply impossible for most of human history. In fact, what's incredible is that doctors and scientists could even start fighting viruses before they knew what they were. So Edward Jenner in the late 1700s created a smallpox vaccine, which was actually consisted of cowpox viruses that he would administer to people, and that would actually create a protective immunity, not just to cowpox, but to the related smallpox. Jenner had no idea that he was dealing with two different kinds of viruses at all. In fact, the word virus was used when he was creating the vaccine. That word did not really exist. In fact, it wasn't until the end of the 19th century that scientists got their first real inkling of viruses. And they did not come from people or from dogs, but actually from tobacco. Tobacco plants have a disease called tobacco mosaic disease. You can see here why, because it creates this mosaic pattern. And tobacco farmers were, to them, this disease was the worst enemy. And so, with the rise of the germ theory of disease in the 1800s, they were wondering, well, could this be a germ that causes this disease? Could it be bacteria, for example, or maybe a fungus? Well, a scientist named Martin Weigeri did a very interesting experiment. What he did was he took a thick tobacco leaf, and he mashed it up in water, and he poured that fluid through a porcelain filter. And the holes in the filter were so small that the water could get through, but bacteria and fungi could not. And yet, when he took what seemed to be just pure water and injected it into another leaf, he made that tobacco plant sick as well. And he could do the same thing to that second plant. He could pass it through a filter, and it could cause an infection in the next one. So Weigeri argued that there was something infectious that was causing the disease. It wasn't bacteria, it wasn't fungi, it was something incredibly tiny enough to get through the pores, and it could pass through a filter. And he picked out the name virus, for whatever this thing was. Virus is the name has very old history, but it means things, for example, like a poison, or even a seed. He used this word now at the end of the 1800s specifically about whatever it was that was making these tobacco well. So, for example, rabies. They found the rabies virus very quickly. They were able to isolate the influenza virus by the 1930s. So they were starting to see that different species, in fact all species of animals, plants, and bacteria can be infected by viruses. But they would use this filtering to find it. Because they were just so small, too small for the best microscopes of the day. The first step towards seeing these viruses actually happened in the 1930s when a Nobel Prize-winning scientist, Wendell Stanley, created a huge batch of tobacco mosaic virus, that thing that makes the tobacco leaves sick. And then what he did, he basically effectively sort of cooked it down so that eventually all he was left with were pure virus. And in fact, what the viruses did was they locked together and crystallized. In other words, he created virus crystals. Now you can make crystals out of salt, you can make even crystals out of protein. You can't make a crystal out of tobacco plants. You can't make people into crystals. This is the kind of thing you do with inanimate matter. And if you were these viruses that he could crystallize, this is a picture of these crystals. And Stanley could put these crystals, up on a shelf, like salt, for months. And take them off the shelf, and he could throw them in water, and now he had viruses that could infect tobacco plants, just like viruses had done decades earlier. So, when he published these results in the 1930s, it's really hard to convey just how much this blew people's minds. And one of my forerunners at the New York Times in 1937 wrote a piece, and the headline was "Viruses and Life." And this person wrote, "The import of these researches staggers the imagination. Viruses are neither dead nor alive. They seem to lie on that shadowy borderland where matter is ready to leap into life." In fact, this was so mind-blowing that another reporter came back to it in the 1940s, asking the question, "Can crystals live?" Kind of a question that not many people had asked before. When Dr. W.M. Stanley of the Rockefeller Institute's Princeton Station crystallized the virus which produces the mosaic disease of tobacco, there was a great hullabaloo among the biologists, and rightly so. Were these crystals alive? Apparently no more so than diamond-clad sands or other crystals with which we are familiar. Yet when virus crystals were put on tobacco leaves, the mosaic disease spread like a slow fire over a whole field, just as if it had been infected by living bacteria. So if you look at the way that these particles kind of set up life, it's very hard to figure out what viruses are. On the one hand, you've got life where you have things that are growing and have other features in common. And then on the other side, you have not life. You have a diamond. And you have these virus crystals. What they are when they're not crystals is hard to say. So this line, this borderland that reporter called, that is the subject of my new book, Live Sex, The Search for What It Means to Be Alive. This is the lovely British cover. The British edition has come out recently. And viruses play a big part in my book. I talk about lots of other things. I talk about when our own lives begin and when is the point that I die. I talk about life on other planets. I talk about strange animals like tardigrades in soil that can basically turn into glass. When you're in these strange borderlands, it's a weird place to be. And this is very much the home of the virus. So I'm going to talk today particularly about viruses. But first I want to sort of bring you up to speed from the 1930s. We know a lot more about viruses now
there.
And it turned out, as scientists put other viruses under the microscope, that viruses have lots of different shapes. So, for example, some of them look like peppermint. Ebola looks like a snake. Adenovirus to me looks like kind of like a satellite. ES virus, which infects bacteria, that looks like, I don't know, looks like a lunar lander. Viruses come in a assay made of shapes and sizes. And yet, we call them viruses because they have certain things in common. Rosalind Franklin, who is perhaps most famous for her work on the structure of DNA, actually before she did that DNA work, she spent a lot of time looking at this tobacco virus. This became one of the most important viruses in the history of science because scientists could build on the work of previous scientists and really start to get the first good look at any virus and then understanding what made it up. And what Franklin's work was really important to help us understand was that viruses are really, really different than cells. In the sense that here you have tobacco mosaic virus, and the blue and the violet shapes there are alternating proteins that kind of get wrapped around like a Napoleon pastry. And sandwiched inside of them is a genetic molecule. Our genetic molecules are DNA, which is double-stranded. A lot of viruses use RNA, which is single-stranded. And so that's wound inside of this protein shape. So this virus doesn't have any enzymes inside of it. It doesn't have like a ribosome, for example, which is a factory for building new proteins. It doesn't have any of that. It's just the shell and the gene, and that's it. So Peter Medawar aptly called a virus a piece of bad news wrapped up in a protein. And despite the different shapes that viruses have, this is the influenza virus, for example, that same structure is true for all of them. So again, here you have a flu virus with a round shell, which is studded by proteins, and then inside of that you have RNA, which is actually little segments, as opposed to being like a continuous strand. The general way that new viruses come to be is pretty much the same from virus to virus, and again, it's different than cellular life. A virus itself does not grow and divide and become a new one. The viruses do not mate and have little baby viruses. What happens is the viruses latch onto cells, they fuse, the genes go into the cell makes proteins from those genes, it makes new genes from the virus, and then new genes and proteins are packaged into new viruses that escape the cell. That's it. That is the virus life cycle. We've gotten to the point now that it's possible to take a virus like this, SARS-CoV-2, the cause of COVID-19, and in very little time figure it down to the atomic level. This is actually a model that Romeo Amaro and her colleagues put together at the University of Colorado, San Diego. This is a model they're building down to every single atom in the virus. And so what you're seeing is that it has this round shape, and then it has these one kind of protein that doesn't surface. This is called the spike protein. You've heard of the spike protein because this is essentially the key that the virus uses to unlock our cells. The spike protein, as it changes shape, it is able to attach to certain kinds of cells in our body, first in our nose, and then in our lungs, and then in other organs. And then it can fuse and dump its RNA into its host cell. And then that cell then makes new coronaviruses. So here we've gone from, viruses as this deep mystery of why God become landed or why we get sick with the flu. We've gone to viruses simple fundamental question. Now that we know about viruses at the level that we do in 2021, are viruses alive? No. What does it mean to be alive? I think we'd all agree, for example, that this snake that's wrapped itself around my neck is alive. Why? What about it means that it's alive? It moves. Okay, movement is interesting. Robots can move. It can eat. It can metabolize. That's very important in terms of life. All living things have to get food in one form or another somehow, and build organic molecules from them, and break down other ones and get rid of them. The metabolism is obviously something really important to feel. So reproduction is important as well. And there are lots of different ways to reproduce. Maple trees, which I have around my house in Connecticut in the United States, do it in a particular way where pollen fertilizes seeds, and then they produce these beautiful saguaros, which then fall all over the place. They clog my gutters, and they sprout up into little volunteer saplings all over the place. Every year, a single maple tree will just churn out thousands of these things, although the vast majority of them will not survive to be able to reproduce themselves. But some of them do, which is why we still have maple trees around for millions of years. Another feature of life unites us with things that at first might not look like life. So this is a slime mold. It's a giant single-celled organism that lives on the forest floor. You can see them for yourself with your naked eye, and they grow in quickly. If you're wanting to know what to look for, the name of one species is dog vomit, so that kind of gives you an idea of what to keep your eye out for. This is a slime mold called Ficera, and like other slime molds, it's just a giant cell that sends out tentacles to find food. In this case, the food is oatmeal, or more specifically, the bacteria that grows on the flake of oatmeal. You can see one in the lower left-hand corner that the slime mold just reached. So as the slime mold is going out and searching for food, and sending out all these tentacles, as it finds food and is digesting them, it keeps reworking its network of tentacles. Amazingly, it can create a network of the shortest distance to connect these different pieces of food. It's like solving a mathematical problem. So here you have something that is taking in information from its surroundings and then coming up with behaviors to advance its own survival based on what it learned. So, scientists who study slime molds will say, well, this is intelligence. This is intelligence not in a sort of test-taking way, but an intelligence in a more fundamental way, fundamental to life. What does that mean? What does that all add up to? This is a question that is not at all new. This is a quote from 1704 from a chemist in New York, Stahl, who was wrestling with how to make sense of the scientific revolution's innovations in terms of physics, in terms of thinking of the world as matter and motion, and trying to understand, what makes life different? Do you have a feeling that life is different from other things? So yes, above all else, when we come going to this, know what is life. Now, in my book, I demonstrate that there's not really a good agreed-on answer to that question. There's no standard definition of life. Here, imagine, for example, not having an agreed-on definition of an electron or of hydrogen. Yet somehow biologists manage to do lots of important work without actually agreeing on what it is that they're studying. One of perhaps the most popular definitions, that doesn't mean it's the right one, but the one that you see very often is sometimes called the NASA definition of life. This is a definition that they had to agree on what it is they were looking for. What's the point of sending a really expensive spacecraft and leaving people to go look for life when you don't even agree on what it is that you're actually looking for? So we need a working definition, something that could advance research. And so this is the definition I came up with. Life is a self-sustained chemical system capable of undergoing Darwinian evolution. Nice, as a writer, I appreciate it. Like it does a lot with a little. So let's think about viruses according to that definition, just to start the conversation. And so I want to focus on the last part of this definition, capable of undergoing Darwinian evolution. And I think we are living through that definition of life for viruses right now with the pandemic. When the pandemic emerged in late 2019 in China, scientists were quickly able to isolate the virus SARS-CoV-2, which causes COVID-19. But the spectacular pace at which it took over the world and has turned humanity's collective existence upside down has a lot of people asking, where did this come from? How did this, we didn't know about this virus before 2019. How is this possible? And there have been speculations that maybe scientists made it in a lab. Scientists I talked to didn't see very compelling evidence for that. Instead, they argued that most likely what happened is that it spilled over from a virus that was infecting animals, specifically bats. And the evidence is all of the coronaviruses that infect bats today. We just scratched the surface of the huge diversity of coronaviruses that infect bats, and you can draw a family tree of them. This is the kind of evolutionary tree that Darwin drew when he was developing the theory of evolution. Except now we can show with them how coronaviruses have spilled over. So in 2002 we had SARS, caused by another coronavirus on a different branch, and more recently SARS-CoV-2 has spilled over. Coronaviruses can also go from bats and other animals as well, like pangolins or mink and so on. So, the details of precisely how this virus got from bats to people, there's going to take years of research.
Det står nicht zu fassen.
The broad outlines are pretty clear here, that over the course of centuries, coronaviruses have been spilling over from bats, perhaps other species as well. Now, once this new coronavirus got into our species and started spreading from person to person, evolution did not stop. This is a family tree of SARS-CoV-2 just from people, and you can see how it's branching out into these different lineages. And these little dots mark mutations that accumulated in those lineages. So you have this raw material in terms of new mutations that can make evolution possible. First really clear evidence of this came at the end of December when a new variant, Z117, Alpha, took over Britain. Suddenly, this variant was dominant, and soon it was spreading to other countries as well, becoming dominant there. What made it dominant? Well, this family tree here shows a whole bunch of mutations to proteins like the spike that I mentioned before on the surface of the virus, which appear to have made them more fit. In other words, these viruses may have been able to evade our immune system better. They may have been able to potentially be able to grab on more tightly to cells. They had an evolutionary advantage. And so, by looking at these mutations, scientists would try to understand how it was that Z117, Alpha, was wiping out all the other variants. And then in turn, Alpha has been wiped out by another variant, Delta. So we're seeing wave after wave of natural selection, Darwinian evolution, happening to no one has ever found a virus that has like a full complement of machinery for these things inside of the virus, inside of what's called the virion, this shell inside. So, a journalist once asked Gerald Joyce of the Salk Institute, who was one of the people who came up with that working definition, the NASA definition of life, about viruses. And he said, according to the working definition, a virus doesn't exist. In other words, a virus is not life. A lot of people who agree with him, many virology taxonomists say that viruses are not living things.
Oh.
But there are others who disagree. One of them
Keep this on me!
who has been arguing for this in the past decade is a scientist named Patrick Forterre. And he doesn't use the NASA definition of life. He argues, here's a passage, life and living processes are simply names for complex evolving forms of matter that are now present on our planet. And so, in this view of things, he welcomes in virus to life and living processes. Forterre has championed an idea called the virocell. According to this concept, when a virus gets into a cell, that cell is no longer what it was. And so you have two different kinds of cells, both of which are alive. The virocell is making viruses as opposed to the uninfected cell. He calls that a ribocell, named after the ribosome, which is that factory I mentioned that produces protein. So that virocell, that is the incarnation of the virus to Forterre is most important. And he argues that it is alive and distinct from it. This is a wonderfully complex diagram from a recent article in Nature, which kind of shows you some of the key steps in SARS-CoV-2's life cycle. You know, it's the same basic cycle as I told you about before. The virus goes in, cell makes new viruses, new viruses come out. But what I wanted to focus on here, amongst all the details of how this works, is this change in shape, this change in structure that happens in the cell. You see these bubbles here? This is a wild and amazing feature of an infection with SARS-CoV-2. These bubbles fill up the cell. Like the cell is not doing much of its old work anymore. It's not maintaining its own structure. It's building new things. This is a close-up microscopy of those bubbles. There are double layers. So here the layers are in yellow and blue, and those green things inside, those are all individual pieces of RNA. Those are viruses, new viruses that the cell is making. How they're getting into these little vesicles, these little nurseries? Scientists don't know how they're getting in. They don't know why they have to get in there. But it's an essential part of that life cycle of the virus, that the cell has to become this virus factory. The little red things on the side of the vesicle you see in the lower left, that's a channel. That channel is encoded in a virus gene. And that appears to be either letting the RNA in or out of these bubbles. Still not clear what. But this RNA that's being stored in these bubbles is then being used by the cell to make new viruses. So this is really, SARS-CoV-2 is showing us Forterre's idea of a virus cell in action. And what that virus cell is doing is it's making new viruses and new virus cells. And so the way Forterre puts it is, whereas the dream of a normal cell is to produce 2 cells, the dream of a virus cell is to produce 100 or more new virus cells. Carey has a wonderfully poetic way of conveying his ideas. Some scientists think that he takes a little too much poetic license, but certainly as a writer myself, I appreciate it.